happy now to introduce Barbara Hahn, a disease ecologist at the Cary Institute of Ecosystem Studies. Barbara believes that deep collaborations between technology and biology can lead to a world where infectious diseases no longer takes us by surprise.
Good day, everybody. My name is Barbara Hahn. I am an infectious disease ecologist at the Cary Institute of Ecosystem Studies, which is in New York. So it's very early in the morning here. I would love to share with you today some of the work that my group has been doing over the past several years, trying to push forward this very important field of infectious disease intelligence. And in this work, we essentially try to leverage the power of the big data that are available for biology and ecology and environmental science and combine them with machine learning approaches in order to make better predictions about infectious disease. And I'm going to unpack a little bit about what I mean by those phrases and also provide some examples for you today about what that looks like in practice. Let's see here. Okay. So I think perhaps now, more so than in any other time in history, we are very familiar with the numbers of zoonotic diseases or infectious diseases that originate in animals, such as mammals, and spill over and transmit to cause disease in humans. The majority of these infectious diseases come from vertebrates and animals. The ones that are emerging in humans today, 2/3 of them come from mammals. And several years ago, a couple of colleagues and I did this very broad baseline review just to try to understand where the diseases are in which mammal species across the world. Nobody had done this before, and the data are available, but of course, as most of us are familiar, it takes some doing to collect those data and put them all into the same place. When we did this analysis, we discovered that there are hundreds and hundreds of species of mammals out of the total of 5,000 that we have available today, that we know about today. And they carry many zoonotic diseases, some of which are not causing huge problems in humans, but have caused disease through, once or twice spillover events. And those are in comparison to others such as SARS-CoV-2, which we now know has the potential or has become pandemic. And the worry, of course, is that there will be others like SARS-CoV-2 that even if they don't have the potential to go pandemic, they still have the potential to cause great economic and human losses globally. So I think right now what we are seeing is our human state in this emergency status. So if you think about the risk of infectious disease, you can think about it in terms of a watch phase, a warning phase, and an emergency phase. And in the emergency phase, what we're dealing with is containment. We have a wildfire on the landscape, and we need to contain that damage. And so our response to that outbreak is fast-paced, it's right now, and it's trying to put out a fire that's threatening to overrun our ability to contain it. Now, when we are not in an emergency situation, what we tend to fall back on is this warning phase. And in this phase, we are really just dealing with pathogens that already have names, that are known risks, and that we need to understand more about them in order to do a better job of containing them or preventing future spillover events. And this is where the majority of our research efforts lie. It's where the majority of our peacetime efforts go. But upstream of this even is this watch phase, where prevention is the key. So in this phase, what we're really concerned about is those changes to the environment that we are making, changes to the global climate, changes to the way that we are living our lives, for example, being better connected through transportation. And in these situations, what we're worried about is new spillover events or the evolution of new pathogens in animal populations that are experiencing rapid global change. And so the viral evolution in these species is different, and the contact patterns between these organisms and humans is different and could cause risk in the future for a spillover. What we don't have a good handle on is how to do research in this watch phase. And this is where my lab has really situated itself, trying to combine the data that exists across the watch, the warning, and the emergency phases with analytics approaches in order to make predictions that we can act on, predictions that help us to be better prepared upstream, and predictions that we can take to improve our policies and improve our management and the way that we live on this planet. So I'm going to give you a couple of diseases, a couple of examples about what I mean and for particular diseases. When I talk about predicting zoonotic disease, what I really mean is predicting what particular species these diseases come from. So who, where these diseases are most likely to arise, what environmental signals cue us in to when these diseases are going to spill over or increase in incidence in a human population, and ultimately why. Why are we seeing these changes now, and why are we seeing increasing emergence in human diseases coming from animals? And to do this, we combine machine learning, like I said, with data that describe hosts, so mammal species, like I mentioned before, and I've just pictured here rodents and bats as two of the most species-rich examples out of the whole mammal group. And we apply them to vectors such as ticks and mosquitoes. And we also apply them to particular pathogens. So we like to think about zoonotic pathogens as a giant group, but we can also kind of dive deep and think about things like Ebola virus or other filoviruses. We can think about things like beta coronaviruses such as SARS-CoV-2. So I'll just give you a quick couple of examples here for predictions that we have made in the past. The first time that we had applied machine learning methods to make predictions about what rodent species, for example, should be able to carry zoonotic diseases, our models performed with very high accuracy. So this is just a map that I'm showing you of the known information. So we have described so far on the planet about 2,277 rodent species, or individual species, and about 10% of them are already confirmed to carry at least one zoonotic pathogen, up to about 13 different pathogens in a single species. Now, when we train our model, what we are aiming to do is help it to recognize the characteristics of a species that is a good carrier for a zoonotic disease. What is it that makes these species good at carrying one or multiple pathogens? And what are those species, what do those species have that make them special and different from the ones that don't seem to carry any diseases and don't cause any problems to humans? And in order to train these models to do this, we give it lots of data that characterize those species. You can think about it in terms of when we go to the zoo or when we are looking at a bunch of rodents on the landscape, we can, with our eyes, see that they are different, that they have different characteristics. They sort of have a different life strategy. Some things have babies very quickly, starting at a young age, and other ones sort of spread out their reproduction over a long lifespan and invest their resources differently. And so I'm just showing you a couple of examples here in different categories like size and reproduction and where they are located across the landscape that give us a sort of caricature of what these species are and how they're different from each other. And the reason this is important is because mammals in general have evolved over a long evolutionary history to deal with environmental pressures. So this includes things like predation and being able to get enough food to feed themselves, but also things like pathogens and parasites, which have always caused a huge selection pressure on these species and are something that they have had to adapt ways to deal with as they have evolved. And so these traits provide a snapshot of what a successful species looks like that is able to contend with these different selection pressures, including parasites and pathogens. Now, when we trained our models to distinguish reservoirs, so those species that carry lots of diseases, from non-reservoirs, or those that we don't know carry anything, our model predicted with very high accuracy. It was upwards of 90%. And it made also some very specific predictions. The thing that's different about this approach is that it not only gives you a sort of trait-based and biologically well-informed, data-driven way of making predictions about what distinguishes species, but it also gives you a rank-ordered list identifying species that seem to have the same characteristics as a reservoir, but have not yet been described as carrying anything. So in a sense, it gives you a surveillance target. Here is something that looks like it should carry diseases. It looks just like the other zoonotic reservoirs, but we don't have any information on it. Maybe that should be a surveillance target. So when our model performed, this was back in 2015, it identified a couple of species, which I'll just list for you here. This one is the Onychomus leucogaster. It carries now, we know that it carries plague and Bartonella. So these have now been validated by independent field studies. And here's another one in the Eastern Hemisphere, very widespread, the Microtus gregalis. It has been confirmed now to carry both plague and a hemorrhagic fever. And just to give you an expanded example here, we also did the same analysis looking specifically at Ebola virus and trying to understand what are the bat species that have the capacity to act as a reservoir for these kinds of viruses. And when we did this initial mapping study, we found that our model, again, predicted very well. It performed within the between 80 and 90% accuracy. And here I'm just mapping the top 10% of our predictions from the model. And you can see that in the hot places, there are upwards of 25, 26, up to 28 different species that have a high likelihood of carrying Ebola or very closely related filoviruses, all overlapping on top of each other in geographic area. So if you zoom in on Southeast Asia, this was rather unfortunate, but also fortuitous example of how the models can generate predictions that can then be tested. After about 6 months after this paper was published, there were multiple new filoviruses that were discovered exactly in this region, this hot region. And in addition, the species that ranked number 5 on our list was then confirmed as carrying not only filovirus, but it was a newly described filovirus. This is a newly described reservoir. We'd never seen filovirus in it before. It's not in sub-Saharan Africa. It's in Southeast Asia. And it was expressing a tropism in the lungs, which suggests that the possibility of an aerosolized virus. So the question that these types of analyses give us is, why aren't there more filovirus outbreaks in places like Southeast Asia? And why aren't there more rodent-borne disease outbreaks in hot zones like in Nebraska and Montana in the United States? So in this way, they can identify geographic regions where we should maybe be taking a closer look at what's going on and what changes that humans are making in these regions in order to perturb the natural environment and cause diseases to possibly spill over. So my last example that I'm going to provide for you today is a deeper dive sort of at looking at the cellular level. So when SARS-CoV-2 first emerged on the landscape, we quickly realized how transmissible it was. And because it originated from an animal, we were quite certain that not only is it likely to have a wildlife reservoir, but this pathogen is likely to then become transmitted back from humans into animals because so many people are infected. So our goal in this analysis was really to try to make predictions about where the virus can now become endemic, where can they now hide, because humans are so widely infected across the world. And in order to do this, we had to get a bit creative with our data because, of course, there weren't that many species at the time that we knew were positive or were susceptible to SARS-CoV-2 infection. And so what we did was we went down to the cellular level and we said, well, biology tells us that in order for a virus to in order for a reservoir to become established with this virus, the virus has to bind a cell, and it can't just bind weakly. It has to bind strongly. It has to bind strong enough to get into the cell and then replicate and shed virus into the environment. So there's a whole bunch of downstream things that have to happen, but all of those downstream events depend on this binding strength. So what we did was took the structure of SARS-CoV-2, the spike protein here, and we measured its binding against this ACE2 cell membrane protein. So ACE2 is very widespread across vertebrates. So anything with a backbone pretty much has ACE2 or some version of ACE2. We don't have the structure of every single ACE2 for every vertebrate, but we do have quite a few vertebrates, and all of them are mapped here. So what we did was model the structural binding between those two factors. And we said, well, based on the literature, it looks like that anything that binds more strongly than a cat can shed virus and infect others. And anything that binds more weakly will not be a transmission hazard. So we were really looking at the capacity of these animals to not only become infected, but also to transmit forward, which is the thing that is risky for humans going forward in this pandemic. So when we zoom in on the mammals are the ones that we are most concerned about, basically because we domesticate them, we have them as pets, we have closer contact with them, and we also had more trait data available and more ACE2 sequences available for this group. So here, what our model did was it did a great job of separating the blue dots from the orange dots. The blue dots are the ones for which the binding was strong, and the orange dots were the ones for which binding was weak, according to our structural model. And then we asked it to distinguish those species according to their traits. So what are the traits that describe something that looks like it has the capacity to transmit SARS-CoV-2 onward? And the groups that popped out for us were the primates, which is probably not surprising given its close relationship to humans, evolutionarily speaking. There were several rodent species that were looked like they were likely. There are many carnivores, which we now have evidence is true. Their susceptibility does seem to be higher based on zoo records, for example, in which many of the large cats in zoos became infected early on in the pandemic. Perhaps also not surprisingly, there were some major groups of bats that were that looked like they had a high capacity for SARS-CoV-2, and this included the fruit bats, which is the group Pteropus, and the rhinolophids, which are the bats from which the original SARS-CoV-2 virus we think probably spilled over from. There are also a smattering of very interesting other species, which included the pangolins, which of course has strong similarity in certain regions of the genome with the currently described SARS-CoV-2 virus. When we drill down and think about how we did with predictions, here's what we see. We did pretty well with making predictions for a couple of known host species that have now been confirmed. So we made predictions for gorilla and for cougar. Those two have since been confirmed as not only positive, but able to transmit forward. We predicted white-tailed deer. White-tailed deer ranks very high on our list. It is a very widespread species in the United States, in South America, and in Central America. And the white-tailed deer we predicted several months ago as being likely to transmit SARS-CoV-2. And of course, now recent evidence shows that it is very likely to be transmitted between deer and a very high prevalence within the populations, at least here in the United States, at least in 5 states. So we think that it is pretty widespread in deer, and which is a problem for hunting and also for some small farmed populations in other countries. We also predicted the deer mouse, which has now been confirmed by lab work to be transmissible between mice. It's a very widespread mouse in the wild and likes to hang out around human habitations in the Western United States. We also predicted snow leopard, which recently made the news because 3 of them died in a zoo here in the United States. They succumbed to COVID-19 and couldn't be saved. There were a couple of predictions that our model made, though, that have yet to be figured out. So our model predicted that white-footed mice seem like they should have zoonotic capacity for SARS-CoV-2. The lab work has not yet been done, but the white-footed mouse is very widespread here in the eastern United States and is the species that is found commonly around human habitats. In addition, we have cattle and pigs. Cattle and pigs are important because they're domesticated, obviously, but for the United States, at least the lab studies that have been done, pigs seem like they should, according to all of the models so far, pigs look like they should have the capacity to bind SARS-CoV-2 very strongly, which suggests they should also be able to shed virus. But the few lab studies that have been done have not confirmed this. In fact, they don't seem to be shedding infection at all. So that raises the question of why, which I think is a very important outcome of modeling studies in general, because predictions are meant to be tested. And the gold standard for prediction is lab work. But lab work is extremely expensive. And so having this nice iteration between models that give you a prediction for particular species in particular regions, and the field work that's done to validate whether that's true, and then also the lab work to show how it's done and why the predictions aren't coming true. That sort of feedback loop is missing currently in pandemic intelligence. Cattle, I believe, do become infected, but there's not evidence to show onward transmission yet. I'm showing you a map here of the top 10% of species that were predicted to be positive for SARS-CoV-2 and to transmit it onward. You can see that the map is colored, pretty much the entire map is colored in. But of course, that's not the most important thing for transmission. The most important thing for transmission to happen is for there to be a contact event such that spillover happens. So humans are transmitting it to each other. Animals are going to get it to humans when humans spill back into animals. So really the places that we're worried about this happening are places where animals don't mind overlapping with humans, and in places where humans are highly infected. So over 100,000 people minimum infected with COVID. So when we subset the map that way, we find that there are a couple of species that don't mind being around humans and also overlap in COVID-19 hotspots. And that includes the white-tailed deer, the red fox, Vulpes vulpes, which is extremely widespread across the world, the agouti in South America, and an antelope in Africa. And these species are just 4 examples that I've cherry-picked because they're the most common. But I think that this example is a very nice one for how we can leverage machine learning approaches, not just to species level traits for predicting species, but also for predicting viruses using viral characteristics and natural language processing algorithms, or looking at features of vector species, for example, to predict which ones might be carrying the diseases that seem to spill over into humans and we can't figure out where they're coming from. So I think that there is a lot, unfortunately, a lot to be done in stitching together the advances in AI and machine learning and infectious diseases. I want to thank my collaborators on all of this work and my funding sources, and I'd be happy to take any questions. Thank you.
Hey, Barbara.
Hi.
Hey. I'd like to ask you from the audience. Okay, so based on your experience in this field, how could machine learning help predict? So be happy to take any questions. Thank you. Sorry, there's a petition. How could machine learning help predict, or how could it have predicted COVID-19?
That's a great question. I think it is difficult in forward prediction is always difficult, but I think that there were warning signs early on that this beta coronavirus group was going to be a problem. Of course, I think that one thing I did not mention clearly in my talk is that it's nearly impossible. It's 100% impossible to predict these sort of random mutation events that will lead to a pandemic strain. But what you can do is draw boundaries around the risk and identify where the risk is highest versus where the risk is lowest. I think quantifying that risk is actually the most important thing that we can do. And for SARS-CoV-2, it was clear that the risk was high in some places compared to others, and from some species compared to others, and especially in the beta coronaviruses compared to other viruses. So there were a lot of warning signs, but I think what this example of SARS-CoV-2 tells us is that we need a better mechanism for taking predictions that are being collectively made by the research community and linking them up with surveillance and lab work and doing that whole full circle, which we don't do now. It's just all disjointed.
Yeah, so do you think, since we still don't know the origin of the coronavirus, do you think AI could actually, in the future, understand or trace the origin of the pandemic, like what we are facing right now?
I will say that I'm cautiously hopeful about that. And that's because I know that there are genomic algorithms that are being developed now that are not quite sensitive enough to get to the species level, but they are sensitive enough to say this definitely didn't come from a bat. It probably came from a pangolin, or it didn't come from a pangolin. It came from a cat. I think we're getting closer to being able to do that. We're not quite there yet, but I think in the future, I'm cautiously optimistic that we'll get there.
Nice, hopefully. We have a question of, have you tried to study parrots?
Parrots, like the birds?
Yeah, I believe so.
No, but I would like to.
I believe the question is asked because of their constant interaction with us.
Yes, I think that's a really important point. There are some animals in our predictions that you look at the prediction, you go, oh no, that's terrible. For example, with the SARS-CoV-2 work, we predicted water buffalo, which is extremely widely used, farmed and used as beast of burden in the Eastern Hemisphere. So that point is very valid, whether it's parrots or some other creature. The contact is key.
Mm-hmm. Another question is, how can policymakers benefit from the great work that you do?
I think policymakers have a really tough job, and part of that job requires that they somehow get up to speed on the science, and that's just too much. So I think that if policymakers and scientists were to reach across and try to make connections that are active connections that are aimed at, sort of tracking situations as they unfold, I don't think that continuous interaction is normal. But I think it needs to become normalized. It needs to become the status quo in order for us to be better positioned to make science-based decisions beforehand so that we can avoid catastrophes. But inevitably, catastrophes will happen. And in that case, you still want that connection with science to be strong so that you know exactly what to do quickly. So I think policymakers making an effort, I guess, to reach out to scientists and generate those relationships would be wonderful.
Great. Another question is, do you face any data bias in your field? And if so, how do you tackle it?
Yeah, that's a great question. There is always data bias with infectious disease research because it is extremely biased towards the diseases that humans care about, and it's also biased towards studying animals that are more common. So we generally have more data available for very common species and less data available for rare species. That, fortunately, I guess, isn't always the case because biologists are odd and we like to go out into forests and study rare things. And so in the work that at least in the work that I presented today, one common way that we approach this is to really ask the direct question of whether we are training the models to identify something that carries diseases, or are we inadvertently training the model to identify something that's just been very well studied because it's not rare or because it's in a rich country and there's lots of money for research and all of that. And when we directly ask that question, it seems so far we've never found that the bias extends to the predictions, that in fact we don't have as much study bias as we think we have because we haven't studied the species that are common as well as we should be studying them. In other words, we don't have a lot of data about species in our backyards that we think we should know everything about them because we see them every day. But actually, it turns out that we sometimes have better data for species that are really charismatic but more rare. So I think that the data bias goes both ways. We do have more information about certain kinds of diseases, but not as much information about the species as we think we should.
Amazing. We still have a few questions actually, but unfortunately we're out of time and have to end this session. Thank you so much, Barbara. This has been super interesting. And we'll try, if you can give us some time later on, we can send you the questions if you can answer them.
Wonderful. I'd be happy to.
Okay. Thank you, Barbara.
Thank you very much. Bye-bye.
Bye.